Good morning, dear students. Today we are going to learn some basic concepts and introduction related to pharmaceutical analysis subject. Myself, Dr. Ketaki Dhane, Associate Professor, here with introducing you some basic terms and what will be the part we are going to cover under the subject, that is pharmaceutical analysis. The pharmaceutical analysis is a branch of chemistry which involves a series of processes, includes identification, determination, quantitation and purification. That means in this subject we are going to learn some processes which are deal with identification of any known compound, its determination of the concentration, quantification of that compound and a purification techniques. This is mainly used for the separation of the components from the mixture and for the determination of the structure of the compound. That means here we can separate two more components from each other, and also we can determine what is the structural parameters and the molecular formula and structure of that compound. The different pharmaceutical agents are includes the plants, microorganisms, minerals and synthetic compounds. Importance of this particular subject, that is pharmaceutical analysis. We can identify the drug and which is there in the formulated product. That means we are going to prepare a different dosage form. But if an unknown drug is there, we can identify the type of drug, the name of that drug which is there included in the formulated product. Determination of the active ingredient or the additional impurities. Also we can determine what type of active ingredient is present in that respective dosage form, as well as the additional impurities what are there. We also determine the stability of the drug, the rate of drug release from the formulation. Now we all know the drug when it enters in our stomach and the first class effect, at what rate the drug is released from its formulation. Also we can identify and purify the pure drug from the by using their specification parameters. We also determine the concentration of the specified impurities which are associated with any formulated products, concentration of the drug in plasma or biological fluids. We also determine the pKa values, partition coefficient, solubility of that particular drug compound and stability of the drug which is under the development procedure. So these are all the importance of this particular subject, that is pharmaceutical analysis. Now classification. Based upon the determination type, just now we have discussed all the importance aspects of pharmaceutical analysis, in what aspect it is useful for us. So depending upon the determination of type, there are mainly two types of analytical methods involved in the pharmaceutical analysis, and those methods are qualitative analysis and quantitative analysis. Qualitative analysis is the method used for the identification of the chemical compound. That means if we are going for a qualitative analysis of any compound, we can identify the compound what is actually present in that given sample. And in the case of quantitative analysis method, this method is used for the determination of how much amount of the specific compound is present in that given substance. So basic types of analytical methods. Just now we have discussed about what are the classification points of the pharmaceutical analysis. Now we are going to discuss about the type of analytical methods through which we can identify, we can caliber, we can determine the presence of particular active pharmaceutical ingredient, determination of its concentration, and so on. So these analytical methods are mainly of two types. The first one is classical method. Classical method of determination, identification of any compound is basically divided into three types: gravimetry, titrimetry, and volumetry. The methods through which we can identify the compound, we can determine the concentration, are of basic two types. Classical method, but the first type, that is classical method, is divided into three more subtypes, that is gravimetry, titrimetry, and voltammetry. Now what will be the meaning of the word gravimetry? Gravimetry is the weight of sample which is to be determined after the precipitation. That means we can determine the weight of given sample through precipitation technique. That substance is first of all precipitated through a precipitating agent, and that precipitated substance, we can measure the weight of that precipitated substance, through which we can analyze or we can determine the weight of the sample. This technique is particularly known as gravimetry technique. Next type is titrimetry method. The volume of the solution is determined after the reaction. In this particular type of titration, we can determine the volume of the solution after the reaction. Now, what type of reaction undergone for this particular solute compound? The compound is undergoing neutralization reaction, complex formation reaction, precipitate formation reaction, and oxidative reaction, oxidation reduction type of reaction. We can also call this type of reaction, that is redox reaction. That means when we are going for the volume of the solution or determination of the volume of the solution after the reaction, the reactions are as neutralization, complex formation, precipitate formation, and redox type of reactions. So this is our second type of analytical methods which we are using for the determination under the subject, that is pharmaceutical analysis. Third method is voltammetry method. The volume of the gas. Here we are going to measure the volume of the gas which is to be generated, which is to be evolved by the reaction, is to be determined in this particular type of titration. That means determination of methods under the subject, that is pharmaceutical analysis. The methods are divided into two types: classical method, in which the first method is subdivided into gravimetry method, titrimetry method, and voltammetry method. And the second method is instrumental method. Now the meaning of the word, or the name itself suggests here, that in this type of method we are going to use the instruments. This particular type of method is further subdivided into a electrochemical method. That means instrumental method, also known as electrochemical method. Here we are going to use, for the measurement of that particular compound, we can use, or we can take the current, voltage, and resistance as an basic parameters. So these type of instrumental methods and electrochemical methods are further divided into conductometry, potentiometry, coulometry, and voltammetry. So there are basic two types for the determination of pharmaceutical analytical methods: classical method and instrumental method. Classical method is divided into gravimetry, titrimetry, and voltammetry, but the instrumental method is divided into, instrumental method is also known as electrochemical methods, in which we are going to use the current, voltage, and resistance as an basic parameters for the determination of the concentration, purity, and so on. So these methods, depending upon which parameter we are going to use further for the determination of the concentration of the solute, they are nominated as conductometry, in which we are going to measure the conductance which is flowing through that solution; potentiometry, that is measurement of the potential; coulometry, that is measurement of the current; and the next type is voltammetry. Here we can measure the current as a specified voltage. The next method is in the case of instrumental method, which is basic type of pharmaceutical analytical methods, the second type is optical method. Based upon the measurement of radiations which are to be absorbed or emitted by that particular substance. That means the substance which here we are going to analyze are either eliminating or either absorbing the radiations which are emitted by that particular substance, in which the absorption methods include UV visible, infrared and atomic absorption spectroscopy technique. That means if the compound is going to absorb a particular type of light radiations, they are divided into visible or ultraviolet spectroscopy, infrared spectroscopy, or the third type is atomic absorption spectroscopy. But the next method is emission method, in which the plasma emission spectroscopy, flame spectroscopy and fluorimetry. Next type which is undergoing for the determination of the method of sample is chromatographic method, through which we can determine, we can analyze, we can determine the concentration as well. These methods are high pressure liquid chromatography, gas chromatography, ion exchange chromatography, thin layer chromatography and column chromatography. So these are basic further types of chromatographic methods which we are going to use for the determination of the compound. And the next and some are miscellaneous methods are thermal method, through which we are going to use the temperature as a basic parameter. The thermal methods are divided into differential thermal analysis, thermogravimetric analysis and differential scanning calorimetry. Other methods, these are basic and miscellaneous methods used for the determination of the concentration of the substances: X-ray diffractometry, radioactive method, mass spectrometry, refractometry and polarimetry. Some basic factors which will affect this determination or these methods which we are going to use for the determination of the compound, whether we are using a classical method or whether we are using a subtypes which is undergoing the instrumental method. But there are some factors which will affect the determination of the sample. Now what are those factors which will affect, which will hamper the substance determination either through classical method or through the instrumental method? Type of analysis, whether it is elemental or molecular or atomic or other. So the first factor is whether we are going to use an element for the analysis or whether we are using a whole molecule for the analysis or whether we are analyzing any particular atom at its basic level. What is the nature of that material, whether it is solid, whether it is liquid, whether it is crystalline, whether it is amorphous. This also affects the determination parameters or the method which is to be used for the determination. The precision and accuracy which is required for the analysis of the sample. What type of accuracy and precision we require for this particular compound? The time available for the analysis. The time is very important factors which will be using for the analysis of the sample. Concentration of the sample and the range of that concentration. Depending upon that, we are going to select the solvent range, the solvent which is particularly used for the analysis. So the concentration is also very important parameter. Availability of the standard for the sample. Whether the substance which we are titrating, which we are analyzing, whether that substance standard substance is available or not. That also we have to check before moving towards the particular analytical methods. Availability of the standard sample, and the next is what are the facilities available for that particular analysis of the sample. Now we are moving towards the actual type of titration method through which we are analyzing our particular sample. So the first type and the first method is titrimetric method. Now what is this titrimetric method? This method is a volumetric procedure. What is involved in this procedure? Through which, that is a volumetric procedure for the determination of the concentration of the sample by addition of known concentration or volume of the standard substance. So what is the meaning of the word titrimetry? Here we are going to use a volumetric procedure. That means some amount of volume we are going to check, or we are going to take for the determination. So here we are going to determine the concentration of the sample by addition of a standard solution. Standard solution means what? Whose concentration is known to us. That is known concentration or known volume of the standard substance. If we are using a standard substance or standard solution for the determination of the unknown sample, this technique is known as titrimetric method. This reacts quantitatively with the sample solution. Then a chemical substance which is used to detect the end point by the color change method or by the precipitate method or by the complex formation at the equivalent point. That means if we are going for the titrimetric analysis, we are going to use either a color changing agent, that is indicator, or the precipitating formating agent or the complex forming substance, through which we can determine the equivalent point of that particular titration. So the compound which we are using for the change in the color is known as indicator in the case of titrimetric method. Now the general terms which we are going to use for the titrimetric analysis. The first and foremost very important terminology is titrant. This is a solution of the known concentration of standard substance. So what is the meaning of the word titrant? The compound or the substance whose concentration is known to us is known as standard solution. Titrant. Titrant means whose concentration is known to us. Titrant means this is a solution of the unknown sample whose concentration is to be determined. Means the solution whose concentration we are determining through this whether the classical method or whether the instrumental method is known as a titrant. And after the completion of the reaction, the point which we will get is known as equivalence point. We will discuss the basic definition. This is a point where the reaction between the titrant and the titrant are completed. So it can be detected by the color change of the indicator which we have already used or already added in that reaction mixture. So these are very, very basic and important terminologies which are required when we are going for any titrimetric analysis. Now, what are the types of titrations? The types of titrations can be classified based upon the parameters, that is measurement, nature of the titrant used, and so on. So till we have discussed what are the methods used for the analysis of any pharmaceutical substance, what are the importance of the pharmaceutical substances, and so on. Now we are moving. What are the types of titrations? Just now we have discussed what is actually a titrimetric method, some basic terminologies, and now we are moving towards the types of titrations which we are using while we are going for any analysis of the pharmaceutical substance. So the first is the type of titration based upon the measurement. First is volumetry. The measurement of the volume of titrant required to complete the reaction. Here the measurement of volume, how much amount of volume of the titrant we have taken to complete the reaction, is known as volumetry. And what is gravimetry? The measurement of weight of the titrant required to complete the reaction. So these are basic two methods of types of titrations which are depending upon the weight or the measurement of weight of that particular substance. The next type is based upon the nature of the titrant. What is the nature of that titrant? So depending upon that, we are going to select a different method for the analysis of the sample. So the first method is aqueous titration. What is the meaning of the word aqueous titration? Aqueous titration is the titration based upon the titration of the sample by using aqueous titrants. Those are hydrochloric acid and sodium hydroxide. So if we are using the aqueous titrants like hydrochloric acid and sodium hydroxide during the analysis of any compound, that particular type of titration is coming under the category of aqueous titration. Next is non-aqueous titrations. So the non-aqueous titrations are based upon the titration where we are going to analyze the particular unknown compound or the analysis of that compound through non-aqueous titrant. Here, instead of using hydrochloric acid and sodium hydroxide, we can use a dimethyl formamide as an non-aqueous agent for this particular type of analysis. Based upon the principle of the titration. So first type is depending upon the weight of the measurement or what is to be measured, that is weight. Then next is depending upon what is the type of the titrant, whether it is aqueous in nature or whether it is non-aqueous in nature. The third is based upon the principle of the titration. During titrimetric procedure, the compound is following what type of principle during the reaction mixture. This is also divided, that is acid base titration. This is a type of titration based upon the titration of either acidic or basic compound by consequent acids or bases. So here we can take an example of titration of hydrochloric acid with either sodium hydroxide or a titration of the potassium hydroxide. Both of them are the strong bases reacted with a strong acid, that is hydrochloric acid. Based on the acid or base to be neutralized, again these reactions are classified into two subtypes, acidimetry and alkalimetry. Whether we are using acid or whether we are using base for the reaction, they are further divided into, or the terminologies used for those are acidimetry and alkalimetry. Titration of free bases or salt of weak acids with a strong acid is known as acidimetry. Here we can take an example of titration of strong base with a strong acid, that is hydrochloric acid. Alkalimetry, titration of free acids or salt of acid with a weak base or with a strong base is known as alkalimetry. Now, what are the basic applications of the techniques or the types of titration, that is acidimetry and alkalimetry? We can determine the alkalinity determination of the water. If we are going for the analysis of potable water and so on, so if we want to check the alkalinity of that particular water sample, we will go for the acidimetry or alkalimetry type of titration method. We can determine the acid content of wine as well as fruit juice. Determination of the acid content in the milk is also determined through acidimetry and alkalimetry technique. Determination of the total acid number, total base number in all the petroleum products is also determined through acidimetry and alkalimetric method. Determination of the boric acid in the cooling fluids for the nuclear power stations. Determination of the free or total acidity of the planting baths. Active ingredient in the drug or the raw material for the pharmaceutical industry. And very basic and very important application is determination of the total nitrogen determination through Jadal's technique also uses a principle involved in the acidimetry and alkalimetry technique. The next and very important type of titrimetric method is oxidation reduction titration. These reactions are mainly based upon the oxidation reduction reactions by using oxidizing and reducing agent. For example of this particular type of reaction is permanganate type of titration. These reactions are commonly known as redox titration. By the name itself indicate the change of the oxidation state or the transfer of the electrons of the reactant by the use of oxidizing or reducing agent. Oxidizing agents are, for example, iodine, potassium dichromate, potassium permanganate, cerium salts, hydrogen peroxide, oxidized chlorine, and so on. Some of the examples of the reducing agents are sodium thiosulfate, oxalic acid, ammonium ion, Mohr salts, hydrogen peroxide, and phthalate oxide. So these are some of the examples of the oxidation reduction, that is redox titration. Now we are moving towards the some basic applications of the redox type of titration. Through these techniques we can determine the chemical oxygen demand of the water. We also determine the oxidation capacity of the water by upper magnet method. Determination of the free and total sulfur dioxide in the water, wine, alcohol, dried fruits, through which by the redox type of titration. Determination of the vitamin C in any compound, titration of the copper and tin using iodine through redox titration, titration of the chromium, and determination of the water in the hydrocarbons. Our next type of titrimetric method is complexometric titration. These titrations are based upon the complexion reaction by using a complexing agent as an titrant. EDTA titration. These reactions are carried out by a complex formation reaction by combining ions using a complexing agent like ethylene diamine tetraacetic acid, that is EDTA, and the end point which is detected by using metal ion detector. So what is the meaning of the EDTA titration? In which these type of titrations we are going to use EDTA as an complex forming agent for this particular type of reaction, and by using the endpoint is detected by using some metal ions in this type of titrations. Some basic applications of the complexometric titrations are determination of hardness of water, determination of presence of some basic metal ions, copper, nickel, zinc, and determination of calcium and magnesium. Our next type of titration is precipitation titration. These titrations are mainly based upon the precipitate formation by using a precipitating agent. For example, silver chloride titration. These reactions are carried out by formation of the precipitate by combining ions or using a precipitating agent. Applications of some of the precipitation titrations are determination of the chloride, determination of chloride in many fisheries products like cooked milk and preservative, determination of the chloride in dairy products, determination of silver in various alloys, for example jewelry and all, and for the titration of halide type of elements we are going for a precipitation type of titrations. So these are all types of redox titration. Our next type is non-aqueous titration. These titrations are mainly based upon the titration by using non-aqueous titrant. For example, titration using perchloric acid or sodium methoxide. Condition required for the titrimetric analysis: the reaction must be simple and easily expressed by simple chemical reaction. Titration should be relatively fast, and the end point should be detected by through very simple physical or chemical change like color change, precipitate formation, and complex formation. Now we are going to discuss a very important terminology, that is what is the meaning of the word standard solution. This contains the known weight of the reagent in a definite volume of the solution. That means what is the meaning of the word standard solution? We can use the known weight of the reagent in a definite volume of the solution. So weight is also known, and that weight amount of the solute we are going to add in the definite volume of the solutions. which has the standard concentration. Through using this basic parameters, that is weight amount of the sample in the definite volume of the solution, is going to have the standard concentration, and the concentration is expressed by the following terms, that is molarity. Concentration of the any substance is determined through following terminologies. The first terminology used for the expression of concentration is molarity. What is molarity? The number of moles of the solute per liter of the solution, which regards to the chemical formula of the compound. Molality, the number of moles of the solute in 1 kg of the solution. That means in the case of molarity, we are going to use 1 liter of the solution with its chemical formula. Molality, we are going to use 1 kilogram of the solution, number of moles of solute which is dissolved in the 1 kilogram of the solution. Normality, the number of equivalent of the solute in the 1 liter of the solution, and equivalent weight is the weight of the one equivalent of the compound. The following standard solutions are used. So the examples of some of these standard solutions are, primary standards are, this is pure form of the substance with the equivalent weight. For example, potassium hydrogen phthalate, succinic acid, sodium carbonate, and EDTA. So these are all the examples of the compound that is primary standard. The primary standard should have the following requirement. If any compound we are using, which must be in the pure form of the substance with the equivalent weight, and some of the examples of the primary standards are potassium hydrogen phthalate, succinic acid, sodium carbonate, and EDTA. The primary standard have some of the following basic requirements. It should be stable in nature. It should be completely soluble in the solvent, given solvent. Its purity can be determined by the standard analytical methods. The reaction with the standard solution should be stoichiometric reaction. The basic terminology used in the standard solution is secondary standard. Secondary standard is the solution which is previously standardized by titrating with the primary standard. Primary standard is a very basic standard solution. Secondary standard is the standard solution which is previously standardized by titrating with a primary standard. Examples: sodium thiosulfate, oxalic acid, and copper sulfate. Indicator: these are very weak organic acids or the bases. Some of the examples of the secondary standards are sodium thiosulfate, oxalic acid, and copper sulfate. Now we are moving towards the next definition, that is indicator. What is the meaning of the word indicator? These are very weak organic acids or bases based upon the hydrogen ion concentration which is present in the solution. The indicator produce different color. These are as follow. The indicator is subdivided into one color indicator, two color indicator, and mixed indicator. One color indicator is perchloric phenolphthalein, which produces pink color. Two color indicator is methylorine, which produces sometime a red color or sometime which produces red to yellow color. And mixed indicator is a natural red or methylene blue. Some of the basic terminologies, that is good manufacturing practices is a system converting the manufacturing and testing of the pharmaceutical dosage forms. This particular method involves guidelines which will determine the quality of that product. Some more basic terminologies are quality assurance. This is wide-ranging concept which cover all the parameters which individually and collectively influences the quality of the product. Quality control is a part of good manufacturing process which is concerned with the sampling, specification, testing with the organization. Calibration. Calibration is the process through which we can ensure whether the instrument readings are accurate with reference to the established standards. This is performed by using a primary reference standard materials for the calibration of particularly that instrument. Validation is the action of checking or providing the validity or accuracy of something which is involved in the pharmaceutical analysis. So thank you all of you for listening. Thank you.